We're talking about predominantly these ones here. When we look at enzyme activity, okay, temperature, pH, and the amount of substrate you have, okay. Cofactors and coenzymes play a part into it because cofactors and coenzymes can go into the enzyme structure and help to facilitate enzymatic activity that way. So if we have enough vitamins and minerals within our diet, we can increase the number of enzymes that we have, and that can increase enzyme activity, obviously. It's not one of the major ones. Again, temperature, pH, substrate amount. Those are the kind of the big three, hint, hint. Okay, so those are the ones that we're going to be talking about predominantly. There are also other different ways that enzymes can be active, and that is because some chemical reactions can produce a stimulatory effect, or they can produce products that can inhibit chemical reactions. So there is a thing that can perpetuate an enzymatic reaction. They can be stimulatory products that cause the activity of more and more enzyme functionality. There's also products that can be formed that inhibit enzyme function and functionality. So it just depends on what the specific thing is. And we'll talk about that in terms of neurotransmitters within the brain. Some neurotransmitters are more inhibitory, where they stop chemical messages from occurring within the brain. So the products are going to be more kind of inhibitory. There are going to be things that happen within the brain that are going to be more stimulatory in nature. So neurotransmitters kind of work in this function where they can either stimulate or inhibit functionality of different structures. So we'll look at E as well in terms of the nervous system. But again, the big three are temperature, pH, and the amount of the substance. The picture says it all here. All right. Enzyme activity increases with an increase in temperature. There's just more bouncing around of these substances. They come in contact with one another a heck of a lot faster when I increase it. Okay. So if I have a lot of enzyme in there, they're bumping around and moving a hell of a lot because of all the heat added into the system. And so as you can see, we get to a point where it peaks out. After that temperature, around 37 degrees centigrade, the enzyme starts to become denatured. And now with the enzyme being denatured, that just tells you one thing. Enzyme shape has changed. Active site shape has changed. So it's not going to bring those specific players together anymore because they're not the same shape. So I diminish the functionality of this enzyme. And as you can see, enzyme activity plummets to near zero. Same thing for pH. pH is basically the same thing here. There is a little bitty window in different regions of the body where enzymes function best within. Okay. And as long as I'm within that little window, I'm going to increase enzymatic activity. These are three different secretions by the digestive tract. Pepsin happening in the stomach, salivary amylase happening within the mouth, and trypsin occurring within the small intestine. And as you can see, each one has an optimal pH for which to function within. Pepsin works best in highly acidic concentrations. So you can see it does best when it's around a pH of anywhere between 1 and 2. If it gets below 1, it you can denature the protein. You can denature the enzyme there. Okay. As well as if it becomes too alkaline. If it becomes too alkaline, pepsin isn't activated as effectively. And so your ability to break down proteins goes down. Salivary amylase works in the mouth. Well, I mean, your mouth is not acidic, at least I hope it's not. Okay. As you can kind of see, it's kind of neutralish, which is fantastic. Okay. Salivary amylase works best around slightly below a 7 pH. Too acidic, too alkaline. You can see the ability to break down carbohydrates is less within the mouth. So what the hell does that mean? It just means you got to have more, there's going to be more stress and strain on the small intestine now. Okay. Same thing for pepsin. If pepsin's not within that little bitty range, you don't break down proteins as effectively within the stomach, and then you're not going to absorb as proteins as effectively within the small intestine. Same thing with trypsin. Trypsin helps to break down proteins, helps to break down some oligopeptides into smaller peptides, tripeptides, dipeptides. Okay. And if it's too acidic, that thing does not function at all. Small intestine is not an acidic place. Okay. It's very alkaline, as you can kind of see. If the small intestine is receiving contents from an acidic environment, what does that tell you about the small intestine? Does that tell you anything about what's happening there from a structure standpoint?
Yes.
Okay, what?
I don't know. I didn't think I'd get this far. I figured someone else would have piped up. I kind of like started it.
You started the ball rolling? You want? Yep.
Yeah, and then I figured, so I was like, I got it.
Yeah.
No. I don't
Nope.
know. It probably means that, a small intestine, if it's too acidic, then trypsin won't work too well.
True, but that tells you right there that the small intestine has to have an adaptation, right? It has to have an adaptation to buffer all that highly acidic chyme coming in from it, right? So they're going to have a number of Brunner's glands there that are going to help to secrete an alkaline type of mucus. There's going to be a number of various different hormones that are going to help trigger mucus production. Sodium bicarbonate to be released into this region of the small intestine, into the duodenum and into the jejunum, to help buffer that high acidic content. Because if it's too acidic, I mean, you can see, trypsin ain't working if it becomes too acidic. And by too acidic, it's just a little bit of, below that neutral position. So it doesn't take much to screw these pH balances that these enzymes function within. Okay. Again, they denature. Okay. They denature. And when they denature, I just change that active site just a little bit. And that renders that enzyme useless and pathetic. Coenzymes and cofactors, they are just vitamins and minerals that add to the enzyme structure that help to get them to become fully active. It is the B vitamins that you think of when you think of these coenzymes. Water-soluble vitamin could be a B vitamin, could be vitamin C. There are a number of water, non-B or C related vitamins that are similar in nature, but these are the B vitamins. Okay, these are the B vitamins. Those are the water-soluble vitamins. Okay, and they help to facilitate, as you can kind of see, the movement of hydrogen atoms, which help a lot of enzymatic function and help a lot of chemical reactions in that regard. Cofactors are metal ions. Okay, they're minerals. They are minerals. There are some examples, okay? If you just remember minerals, I'll be happier than a pig in slop, okay? What's that?
Calcium,
What's that?
magnesium,
What's that?
manganese,
There you go. What we got here?
copper, and then zinc.
Beautiful. Okay. Those are the most kind of major ones that we have here. Help giving rise to some very important, fantastical enzymes that are involved with the metabolic pathway. Okay.
So coenzymes are just vitamins and cofactors are just minerals.
Yes, sir.
Okay.
Yep. And that's all you really need to know. Okay. They help give the enzyme its functionality. Okay. So if you don't have enough vitamins and minerals, okay, if you don't take your Flintstone gummy, okay, then you don't make enough enzymes. You don't make enough enzymes to help make energy or other important enzymes to get things to happen within the body. Okay, so these vitamins, B vitamins, and these minerals are pretty important when it comes to the actual shape and structure of an enzyme. Okay, they can help turn them on and become fully active creatures. I kind of talked about that in that picture where I was looking at these guys right here. Okay. Those are just some words saying the same thing over and over and over again. Okay. Substrate concentration you see gets larger. Small, large amount. Small, larger. Okay. You can kind of see too, there is a rate at which chemical reactions occur just based on the amount of substance. Okay. If a reaction becomes saturated, that tells you that no more chemical reactions occur. So as soon as I get to 100% saturation, it's never 100%, mind you. Okay, it gets to be about 98%, and then chemical reactions really stop occurring within systems. All right, but we get to that maximum rate. Okay, it's that rate that where we can add no more substances because no more chemical reactions occur. All right, that is the maximum rate. All right, we see a number of this type of phenomena within the digestive tract. Okay, in terms of enzymatic function, bringing in specific things into an intestinal cell to get things to occur. When the system has become saturated with too much substance, all the enzyme is used up. So the ability to increase the rate of reaction doesn't occur anymore. Chemical reactions really stop for all intents and purposes. Okay. Oh, I'll just do this here. Okay. I am not going to ask you Goucher's disease. Okay. I'm not going to ask homocystinuria. Okay. I'm not going to ask about these metabolic, these inborn metabolism disorders. Obviously, if a system doesn't, if an enzyme isn't there, okay, the cascade of the pathway for metabolism to occur, whether it's branched or whether it's just a simple, basic metabolic pathway here. If we have enzyme 3 not present, what happens to the final product? Anybody?
You won't get F.
I don't.
Because D is missing.
Yeah. So if I'm missing any one of these enzymes for whatever reason, because I didn't make it, I can't manufacture it, I have some kind of defect in the DNA where I can't form the enzyme properly. If I miss any one of these steps in this pathway here, I can't get to F. Okay. I get C and then nothing else. Okay. I need D. I need E substance to get the F substance. Okay. And that's all they're trying to say here. There are a number of errors that can occur there. And if there's an error, I don't have the pathway. Okay. If I can't make lactase, then I'm lactose intolerant. Okay. Phenylketonuria, the ability to break down phenylalanine. If I don't have it, PKU manifest. Specialized diet, or I have severe intellectual disability. Okay. Albinism. If I can't make the albumin, if I can't make the melanin granules that exist in the cells of the skin, then I don't make any pigmentation. And if I don't make any pigmentation, then I'm more susceptible by those UV rays to cause skin cancer. Tay-Sachs, just accumulation of lipids within the neurons. Again, as you can kind of see down there, that's not a very good outcome. Hypercholesterolemia, just the ability to have too much cholesterol within your blood. I mean, just break down the word, right? Hyper means too much, cholesterol, and then emia is in the blood. Too much cholesterol within the blood. That's because we lack, some people have inborn issues where they lack the protein receptor, excuse me, to bring that cholesterol into the cell for use. And it accumulates within the blood vessels, hardens them up. Okay, so there are a number of errors. I'm not going to ask you this, slide 24. Okay, I'm not going to ask you that. Will I have this image on the exam and say, write out and explain what the hell's happening? No. Could I ask what would happen if in a cascade of 6 different enzymes, if you didn't have the third enzyme, would the system occur? And you would say, heck no. And you'd say, heck no, because you would say there's no third enzyme or there's no second enzyme. Okay, so I can't have the metabolic pathway to occur. And that's all they're trying to show you here. Okay, sometimes I have pathways where the end result causes a negative feedback and it stops the metabolic pathway. There are some pathways where the final product is a positive feedback mechanism and causes the continuation of taking that initial substrate through the process. And whether it's a straight A to B to C to D to E, or whether it's a branched metabolic pathway, it's all the same jazz that you're basically discussing. Does any of this make a great short answer question? At least not these slides. Could I ask you on an exam to describe, I give you a scenario, the pH of this enzyme functions within here. Let's say the pH of an enzyme functions between 6 and 8. You have a system with a pH of 5. Tell me about the chemical reactions that are happening within this system. And then you would talk about how pH affects the shape and integrity of the enzyme, and it denatures it and it renders the system useless. Okay. Could I ask about temperature? Could I ask about the amount of a substance? Would those make great short answer questions? Hell yeah, they would. Those would obviously make great short answer questions. So far on this exam, I have a couple that are pretty decently good short answer questions, right? Mitosis, steps of mitosis and what's happening there, and how enzymes can be affected by a change in pH or a change in temperature or the amount of a substance. Okay. Could I ask about cofactors and coenzymes? Sure, I guess, you would just say these things get incorporated to an enzyme structure and enable the enzyme to be active, i.e. then facilitating enzymatic pathways to occur. Okay, I guess you could talk about those as well. All right. But those make great short answer questions. Okay. That's how I would ask them on an exam, if I was to ask those questions on the exam. What else would be a good short answer question? Exo and endocytosis would be a good short answer question. Okay. What else would be a good short answer question? Well, we haven't gotten to some of that stuff yet. Cellular respiration, metabolism. There's some basic overall concepts that could be some good short answer questions there. Okay. But for what we've done so far, how enzymes are affected, temperature, pH, the amount. Do we have vitamins and minerals in our diet? Again, mitosis. Phases, in the correct order. What's happening in each one? Those make good short answer questions. And so far,
Dr.
that's about
Thorne,
all we got. Go ahead.
question on the pH.
Mhm.
Okay, so the pH basically changes the shape of the enzyme?
Mm-hmm. Yep, because
Okay.
it'll change. Okay. If I just go to this generalized picture here, okay, it'll just change the shape a little bit of this thing. And if you denature it, that's what denature means. You change the shape of the enzyme.
Okay, thank you.
Yeah. Okay. Okay. Let me go here.